sugar. Oh, honey, honey, you are my candy girl, and you got me wanting you. Honey, oh, sugar, sugar, you are my candy girl, and you got me wanting you.
Hey, is this fake guitar, right? From Cake Boss?
Oh yeah, it is, and I'm from the show.
Very nice. I'm glad to be able to play some chord with you guys because this is an extremely complicated case.
How complicated are we talking here?
Well, you see, it's actually two different but related cakes.
Two cakes, huh? Let me bring in another baker. Alright, he's here.
Excellent. So here's the deal. I'm a high school bio teacher, and I need two cakes for my class to teach them about mitosis and meiosis. If I don't use something interesting like cakes, my class will never pay attention.
Tell us a bit about the topic.
Well, you see, I can give you a quick lesson next time we meet, I suppose.
Sounds good, sounds good.
Yeah!
Yeah, baby!
Hey Todd, check out my new breakdancing moves. Word.
He's here.
Oh,
Good afternoon, Cake Boss people.
what's up, buddy?
Okay, so as I said, I'm Jake, and I'm a bio teacher. I would like you to make me mitosis and meiosis cake.
That's cool.
And here I brought you guys a diagram
I love pictures.
of how it works.
Pictures are great.
So I guess first we'll start off with mitosis. And mitosis is the cell cycle, and it's the process of asexual reproduction in cells. Essentially, a single cell can replicate itself over time to produce an identical copy of the same cell. And same the whole time.
Okay.
Nice.
And also, first, the cell replicates its DNA, and in time, through a series of complicated processes, the cell is able to split into two. To learn more, you have to do some more research on your own and use that diagram that I gave you.
So really, I'm just gonna have to do a lot of research to get this cake right.
I guess so.
Now, second, meiosis, a little more complicated. Similar to mitosis, however, is using sexual reproduction. This is crucial difference leads to an assortment of genetic variability. In addition, meiosis produces 4 daughter cells as opposed to only 2. And plus, these daughter cells are haploid, which means they have only half the number of chromosomes of the parent cell. This is because the cell replicates in DNA once, but it splits twice.
Interesting. I guess I'll cover that cake.
And I guess I'll do the mitosis cake.
Thank you, guys.
And goes on the intern? I'll help you with research.
Alright.
Alright, so is this like your teaching PowerPoint?
Yeah, it is. I use it for my students, and sometimes you forget, so.
Yeah.
I guess we can also, while we're filming and making the cakes, we can also describe the process as we go along.
Yeah, that's actually what I was hoping for.
Alright,
Sounds
so
great.
we'll film it and we'll show the process as it progresses through mitosis and meiosis.
Thank you, guys.
Appreciate it. Dab me up, bro. Sorry.
Yeah.
Yeah, he does it. So right now, I'm mixing up the ingredients for this cake. The inside of this cake is going to be like gel-like substance, and that's going to represent the cytoplasm of the cell. Once I'm done mixing these ingredients, I'm going to throw it in the oven, and that's going to represent interphase, and interphase is going to be the longest phase of this cycle. We got to mix the ingredients really nice. So once your mix reaches the right consistency, you're just going to want to take this baby and put it right in the pan. So now you can see my mix has settled, and I also added some icing to represent the nuclear envelope. Now I'm going to throw this baby in the oven. Oh yeah, I feel it. This is going to be the best cake of our entire career. Let's take this, set it down. Set for 30 seconds. We're good to go.
2 hours later.
Safety first. As you can see, the cake has doubled in size and interphase has ended, prophase has begun.
Prophase! As you can see, we use chromosome-shaped icing to show that the chromosomes have condensed around the histones. You couldn't see them before because you can't see uncondensed chromatin. You can also observe that each chromosome has 2 chromatids, and that we also remove the nuclear envelope because we're in prophase. Next, we align the chromosomes along the center, similar to how it would happen in a cell. We also add little pieces of orange icing to represent the centrosomes. These align at each pole and will eventually pull the chromosomes apart with spindle fibers. Just like it would happen in the cell, the chromosomal icing will be pulled to opposite poles by the centrosomes, and the centromeres will dissolve, just like as I'm showing right now. Oh, that's nice. As you can see, the chromosomes are uncoiling into chromatin again, so we got to get rid of those. And the nuclear envelopes form. Finally, the entire cake is pulled apart at this little indentation, which represents the cleavage furrow. As you can see, both sides of the cake are identical. Similar to how, in a cell, the 2 cells produced by mitosis are similar to the parent cell. Joseph! Fan.
Okay, now let's go over to another baker to see his progress.
So my cake will simulate meiosis as opposed to mitosis. It's very similar to Brady's cake, but it has many differences as well. So first we gotta start off by stirring the cake, which I'll use my blender to do. So now that the cake is mixed to the proper consistency, we have to pour it into the pan. Alright, so now that the cake is properly poured, we gotta put it into the oven.
Later.
Joseph?
So now interface has ended, the cake has doubled in size, and prophase has begun.
So now the cake is finished baking, it's going to enter into prophase 1. In meiosis there are 2 phases, PMAT 1 and PMAT 2. Now the cake has entered prophase, we will add chromosome-shaped icing to represent the condensing of the chromatin. In this, the homologous chromosomes will pair up in synapses along the horizontal axis of the cake. One of the homologs will be maternal, one will be paternal. In addition, the homologs will experience crossing over. And the intern here will explain this.
Oh, okay. So crossing over is basically the process in which homologous chromosomes align very closely together and they exchange places with each other. And as you can see right here, they are aligned in the synapses, and basically they are very close together, which allows them to break off pieces and exchange with each other. And the place where this happens is called the chiasma. And this whole process basically leads to genetic variability because the maternal pieces of the allele break off and exchange with the paternal pieces of the alleles. And so basically that's it. Crossing over just basically happens like that and happens in prophase 1.
In metaphase 1, the homologous chromosomes align at the metaphase plate in random orientations, which sets up for independent assortment, as you can see. Okay. In anaphase 1, the homologous chromosomes are separated randomly through independent assortment, which the intern will cover again. The homologous chromosomes are still intact, though. They do not get ripped apart at the centromere.
Independent assortment is basically when homologous chromosomes separate in random manner, and this happens firstly during metaphase when they are aligned randomly in the metaphase phase, and then during anaphase, the homologous chromosomes are randomly placed into opposite poles like that. And for example, the maternal chromosomes can go on one pole, and so can the paternal chromosomes, and that's basically independent assortment.
In telophase, the cell is haploid with a chromatid pair of each chromosome in each pole. At the end of telophase, though, the cell finally splits into two. After telophase, the cell gains a new nuclear envelope on each cell. These cells are haploid because they have half the chromosome number as they started with. After telophase, the cell experiences interkinesis, which is basically just inner phase of mitosis, but the chromosomes are not duplicated. That's it.
Fun.
So after the initial cell is split into two, we have one of the daughter cells right here. This is after interkinesis has occurred and the cell has doubled in size. We have the nuclear envelope is still apparent, and it'll soon fragment.
So basically, after that, prophase takes place. What happens during prophase is the nuclear envelope fragments, and then the chromosomes condense from their original form, which was chromatin, into histones.
After prophase, we have metaphase 2, which is where the chromosomes align to the metaphase plate, and the centrosomes come into play. All this icing represents pieces of the cell.
Okay, after metaphase, move on to anaphase. The centrosomes pull the chromosomes to opposite poles of the cell along the spindle fibers, like so.
And next we got telophase, which is where the chromosomes uncondense and they become a nucleus again. Nuclear envelope forms on either side, since this is broken into two different cells at this point. And then obviously we have cytokinesis, which is where the cell breaks into two. And we have two haploid cells that resulted from meiosis, and there's actually gonna be 4 in total. This is just 2 of them since we left the other one out.
Wow, Ty, you this cake's like rad.
I know it.
Oh my god, this is so
Go ahead,
beautiful.
I'm gonna take that.
Hey,
Joseph!
Joseph!
Where's the other cake?
I'll go unlock the door for him.
Alright.
Joseph! Come here, bring that cake here.
No way.
Nice.
Dude, my students are gonna love this.
Yeah, we're pretty proud of these babies.
Oh!